be really happy about unexpected results. I always tell my PhD student, with every unexpected result, you increase the impact factor of your paper.
It takes time to develop questions and tools.
Creativity is a process and not an event.
Using technology, we are better than the animals.
Collaboration in science is perhaps one of the most important skills for a modern researcher.
Bringing findings early into humans is essential if you do disease research. Never forget about that. And then try to be out of the box, because if you're young, you must be out of the box to replace political weight.
I think the most important thing to have success at any journal when you're submitting is to submit to the right journal and to be realistic about that.
Okay, I will give two examples. Example number one, it takes time to develop questions and tools. And that is just a paper when I started in the group of Stefan Schreiber. That was one of the first genome-wide association scans for non-synonymous SNPs, and it identified a famous gene called ATG16L1. It was kind of at the beginning of what we call the cluster now, and that was published in 2006 by Jochen Hamper, André Franke and the team. And we were also fortunate to take part in that a little bit, and we asked the question, how does it work? Let's look at intestinal epithelia. How about patient-specific signatures that really ended at a question mark at that time point? Let's make a knockout mouse. This one we did. We called the company early 2007. First the S screening, late in 2077. Difficulties, revised the strategy, first mice, later. Yeah. So, and then was still the time where you had to do backcrossing, and it was 1.5 years of backcrossing and so on. So it takes time to develop a model. And at that time, I think we were very fortunate that we could think systematically about patients and tools, how to characterize patients. And it was the first round of the cluster here, where we defined altogether that we need systematic sampling of large cohorts, excellent longitudinal data. There was NGS coming up, right? I mean, it's not that long ago that NGS was not here. We got the first grant for an NGS machine, and something strange, which we then did not follow, and that it's so stupid that we didn't follow. We had single cell analysis ongoing on an NGS machine, but just the wrong model organism. It was Arabidopsis. So like 10 years later, we should have done that in a different way. So then was the first round of mouse experiments in 2010. We teamed up with a team from Boston there, and we had the first hypothesis like 4 years after we started because of the problems we had. And then it took just 3 years to get a nice paper published where we definitely crossed this mouse now to another ER stress model, and then actually showed that if you double knock out these two genes, we have a phenocopy of Crohn's disease, and we understand that there is TNF-dependent transmural fistulizing ileitis. And that was really like from 2007 or late 2006 to 2013. And I think you all have these stories, right? So patients and tools now, not with a question mark, but with an exclamation mark. The second round of cluster was funded at that time. And now we kind of moved still with a patient collection. As you see, the NGS capacity was growing slowly into something that we could call a core facility. There was higher throughput, and there was something that now was called the organoid technology. And we were lucky enough to continue on that story with ATG16, and others did too, of course. But we showed that there is stem cell phenotype in the XBP mice, together with Rick Bloomberg again. We showed age-dependent inflammation in the ATG16 in a nice paper. Again, took 3 years. And then the latest paper from that series is that we really then used the organoid models to show that the true, or maybe one of the main causes behind of that, is that ATG deficient animals have a deficient IL-22 signaling here, and they actually do not react with regeneration, but they react with apoptosis once there is this proliferative stimulus ongoing. So again, now we have the third round of the cluster, and it's still ongoing, right? So now patients are more in bold print. We are much more systematically collecting longitudinal observation cohorts now. We can now do single cell analysis at much higher scale than with these 4 Arabidopsis cells that we had in 2010. We can do crazy things like spatial RNA-seq and metabolomics. And of course, we still continue with the same story. And I think this is science, that you tell a story one by one. So this is the latest paper that we again had in a collaboration with Rick, showing that actually XBP ATG16 now also directs B cells, IgA positive B cell populations, B1b populations in peritoneum. And of course, we still continue, right? This is science. Good. Second example, expect the unexpected. Fostering this was a cluster lecture. As you all know, we have cluster lecture series, and all of you who don't come, please come, because things like this happen here. There was Josef Penninger at a cluster lecture telling, my mice have diarrhea and I don't know why. And then it was a long story that we actually teamed up with Josef and worked for a long while, showing that there is a tryptophan absorption defect in intestinal epithelial cells, leading to the loss of a protective antimicrobial program here. And this leads to a transmissible pro-inflammatory property of the licensed microbiota. And we could actually, in a preclinical model, then show that with tryptophan metabolites, we could cure that pro-inflammatory phenotype. And you see the nice paper coming out of that. And what I not want to show is this one here, but the additional 5 years that it now took Stefan to actually take that knowledge and go into patient cohorts and show that this is also true for IBD. And then, in parallel, also develop a slow release formulation, pharmaceutical and also nutrient formulation of tryptophan metabolites that are patented, where a spin-off company has been founded and where the first intervention now, like 8 years after the initial paper and several years after the patent, unluckily, now goes also to the clinics. The dietary supplement will enter the market in 2020. And just as a side remark, if you really want to endure long preparation times, go pharma with any principle that you will have, this really takes preparation time. So, and last point I want to make, and I think this is also talking about mental health and science and other things, is I think that the scientific competition just got a little bit on the other side and maybe even a little bit unfair. I'll just give you an example, and that's a real example from a conference like 2 years ago. I was sitting in the first row like you here, and then a friend of mine came down, or was sitting inside, and postdoc was giving a talk, and I said, well, this is a really cool and comprehensive story. And he said, well, sometimes you have to stick to ideas. Consumed 4 postdocs. I was like, what? And he said, yes, I mean, it has been 11 years and 4 consecutive postdocs, and only the last one got enough data for the story to be published in Cell. Yeah, and I think this is something that we really have to think about, whether there is a healthy race for increased content in what we do and whether we have enough or too little time to prepare something. And my personal view is science is never linear. Rather looks like this. Yeah. And if you want to do it, you need protected time, no preparation time, protected time for development. I had the luck, right? You also need luck, you need endurance, and you need teamwork. Thank you very much.
Yes, so I got the beautiful task to give a talk about Be Creative. And actually, I'd like to start with a statement that I think creativity is a process and not an event. So no one goes to work saying, oh, today I will be super creative. But usually things start really small and they build on each other step by step. And some years ago, I had the chance to listen to a talk from Bruce Beutler, who got the Nobel Prize in 2011. And I think he summarized this in a really good way, because he said, if you want to win the Nobel Prize with your first experiment, you probably fail. And so I'd like to tell a bit about our story and how we in the end managed, with hopefully also some creative elements, to publish a big paper. And actually my journey started in 2009, when I got my PhD project, Characterization of Aspergillus-specific T cells and Identification of Immunodominant T cell target antigens for adoptive T cell therapy. And so as a young, motivated PhD student, I said, wow, I don't know what that means, but I will do it. And so after reading and discussing a while, the task became more clear. So there's Aspergillus, which is an airborne fungus, so it's inhaled by everyone every day. And this fungus can actually cause detrimental infections, mainly in immunocompromised people, so with 500,000 deaths per year. But actually the rest of the world is quite fine with inhaling this fungus, so there must be some protective immune cells, and my task was now to find these immune cells and antigens responsible for protection. So they're here, so each dot is one cell, so I found around 100 of these protective cells. And I was quite shocked. So what can you do with these cells? And so it was clear that we somehow need to make more of these cells, and then we developed this magnetic enrichment approach. And this really gave us new insight in a lot of different antigen-specific T-cell responses, and I was really quite happy with that. So after having established this assay, so the first results came in, and actually it was a disaster. So the first result was, when we look at these cells, the majority of these cells were actually non-protective naive cells. So we were looking for these, but the majority of them were those. And the second disaster was actually when we further looked at these cells, so up to 30% of them did not only recognize Aspergillus, but they also recognized another fungus, Candida albicans, so they were even not specific. And then the third disaster came when we realized that actually this whole response against Aspergillus is dominated by suppressive cells, so suppressive regulatory T cells. So as you remember, my task was to find the protective immune cells against the pathogen, and actually I found suppressive cells against the pathogen. So after 3 years, it was then clear that Aspergillus does not generate a protective T cell response, and there are no immunodominant T cell targets, and that, of course, was quite sad. But unexpected results let you think out of the box, and I think this is really the beauty of science, that it somehow by itself drives you into creativity, because we generate a lot of unexpected results every day. And the next important point is, I think you really need to follow your results. And as a scientist, you don't go home and don't think about it anymore, but you take your results with you. So you take them for a while down the rabbit hole to think about them in complete isolation. You also take them under the shower, and under the shower is actually a great place to have creative ideas. And one of these under the shower ideas was that I thought about all these regulatory T cells maybe in a different way, and that Aspergillus is maybe not only the pathogen, but maybe something we inhale every day, and so we maybe need more mechanisms of tolerance here. And I can just recommend, don't be afraid of doing experiments that may give unexpected results, so testing whether or not rhinos land on their feet. So for me, that meant not only to look for Aspergillus, but also for some more harmless antigens like plant pollen or house dust mite. And actually, so unexpected results let you think out of the box. And so disaster 1 and 3, we turned into a Cell paper in 2016, where we really showed that these regulatory T cells against airborne antigens are central tolerance mechanisms in humans. And disaster 2, we also turned into a Cell paper in 2019, where we showed that this cross-reactivity to Candida is a major driver of protective and pathologic antifungal responses. And I can just add on to the first talk, so it takes time. So my conclusion of this is, be really happy about unexpected results. I always tell my PhD student, with every unexpected result, you increase the impact factor of your paper. And so that's my summary. Ask questions, do things differently, follow your results, discuss your ideas, and also test your ideas. And finally, I think really creativity is a process, not an event. And again, Picasso said this in much smarter words. I begin with an idea, and then it becomes something else. And I think this is really what's happening in science. So thank you very much.
Collaboration in science is perhaps one of the most important skills for modern researcher. So getting your work published in Nature Medicine or Science, or as Peter showed us in Cell, I think it's more about good data and good knowledge. It's about good communication and collaboration too. So when I started working in Stefan Schreiber's and Andre Franke's lab at Kiel University, so I was greatly encouraged to closely collaborate. I mean, not only with the researchers from the Cluster of Exzcellence, but with researchers worldwide. And yeah, I mean, to create a new way of collaboration, that was basically the idea to establish international disease consortia. So By definition, a consortium is an association of 2 or more researchers, groups, or institutes that have with the objective to pool their resources to achieve a common goal. Here, so together with research groups from 20 more countries, we helped to establish the International Inflammatory Bowel Disease Genetics Consortium. But we also helped to create the International Primary Sclerosing Cholangitis Study Group. But we also joined, as a member, different consortia, for example, the Psoriasis Association Genetics Consortium, or the so-called Eagle Eczema Consortium. Through these consortia, so we were actually able to perform genome-wide studies with tens of thousands of DNA study samples. And I think that was some kind of game changer for many genetic discoveries. And for me personally, it was also very nice a way how to learn a lot of skills from people within the consortia, but I could also share my skills with the people from the same research field. And through this new way of collaboration, we also set the basis to perform projects across different consortia. So I was in a lucky position to lead a multi-consortium project in which we investigated the genetic landscape of several inflammatory conditions, including here inflammatory bowel disease, psoriasis, primary sclerosing cholangitis, and also ankylosing spondylitis. And it was exciting to see that these conditions that affect totally different organs, that they have, or that they seem to have a lot in common on the genetics level. And these genetic discoveries at least partially describe why also, these conditions co-occur in individual patients, but also family members of patients. But so genetics is only one part. We have to find the triggers that lead to the disease in genetically susceptible individuals. And genetics is only one part of the story. So this is why we need you to create new collaborations, not only for genetics, but also for, let's say, other types of molecular data, to harmonize patient information, and so on. And so one last point I would like to make is that I think a new fruitful collaboration can also arise just between a few people from different disciplines. And this brings me back to the Cluster of Excellence here, because I think this is a perfect means here how to generate new collaborations, even on a much smaller scale as compared to these international disease consortia. So actually, a new fruitful collaboration that has emerged from the Cluster of Excellence. These papers here actually are the result of a new collaboration that has emerged from the Cluster of Excellence between me and scientists from the computer engineering department. So a few years ago, there was the predecessor Cluster of Excellence. So we had the idea to program our algorithms on special hardware processors. So we named this way of programming hardware-assisted programming, or simply chip programming. And by doing this, we were able to speed up bioinformatics algorithms. For example, I think many of you know this Plink program. Many of you have used it for genetic studies and so on, to speed this up by a factor of 1,000. And to make a long story short now, through this new collaboration that has emerged from the Cluster of Excellence here, now, two people, to computer sciences, Lars Wienbrand and Jan Kessens, they have moved from the technical department to our institute, and now we have follow-up funding from the BMBF and the DFG. But the initial funding, that came from the Cluster of Excellence. Yeah, and perhaps one day we will also make it into the big journals with our new computational tools here, so that we can reach then a much wider readership as well. So then let's together, let's create data and knowledge that is something besides the parts. And let's collaborate, but don't collaborate too hard. Thank you very much.
I'm a physicist, I'm a technology guy, and I want you now to appreciate the importance of technology. And so actually, what distinguishes humans from animals? A lot of discovery, or almost all discoveries, are actually kind of based on technology. Here's the imaging technology. We see actually at 1600 the microscope that enabled to see first bacteria, electron microscope, you saw viruses and so on. Okay, now coming back to the most important invention is probably the discovery of fire, spending heat and light. And of course, the modern version of fire is the laser. The laser actually can also spend warmth and light. However, it's a type of light not to be found in nature, and therefore we can also use it to measure. And maybe some of you already had the experience with this measuring laser. It can do ranging and determine speed. And this is actually what we do in optical coherence tomography. This is the technology we work on, Lübeck works on for quite some while, for almost 30 years now, coming back to the first talk actually. Okay, OCT is nothing else than measuring the echoes of light. So what you do with sonar or with ultrasound imaging, you do with light. You kind of launch a light pulse on the sample, measure the echo delay time, and that actually gives you an image like here of my retina. It has been commercialized, and because technology is so important, now it's something like 30 million patients are investigated every year. This is one investigation per second. This is the main application for the eye. It's also used endoscopically for intravascular imaging. There it's now almost 1 million patients. So the idea behind it is if you have some lesions, some coronary artery problems, and you are in the cath lab, you actually have this guide wire and angiography is done, and you actually can also produce a optical fiber, and out of this fiber comes the OCT beam, thus performs a spiral scan. You do a pullback, and then actually you can assess this kind of lesions very, very nicely and plan actually the stenting that you implant into the vessel. So this is the procedure. It takes about 3.5 seconds, and the individual OCT image will be something like this, with absolute measurements. Okay, there are more advanced things. So OCT is big in Lübeck, and so you can, because you are very fast now, look really at cilia beading in vivo in real time. You can then fly almost like through a subway through these vessels internally, and you can do live imaging of mucosa and actually even observe this beating frequency of the cilia. Okay. So coming to publications, OCT was first invented in the group of Professor Fujimoto at MIT, 91. So now we have 2020, coming back to this long time it takes. And you see actually like the exponential rise in number of publications. It's dominated by ophthalmology. There it's mainly end users who use it and push the button and do research with the systems and not technology. And you see always when there's a new product coming for a new field in medicine, it actually ramps up more and more. And we see actually here at PMI, we are at the new technologies. So we are considering the 30 years pretty early. There have been bunch of high impact publications. So the first paper from Fujimoto and Eric Swanson and David Huang, optical coherence tomography, has been cited 8,800 times, which I think makes it the 13th most cited paper of science of all times. However, after 2000, actually the number of high-impact publications decreased a little bit. So we cannot actually complain about high-rank publication, but it definitely was a little bit like lowering down. Generally, of course, I wanted to summarize, or I may want to summarize, how it's actually the success of a technology and the age related to, I would say, number of high-impact publications or probability that you get high-impact publications. And I plotted this graph over the technology readiness levels. And usually I think first, if you have a basic technology idea under the shower, of course, under the shower, you will not write like a science paper or a nature paper. But once you do the first like proof of concept, maybe you are here in cloud 9 and promises that may come true and everything is so nice and you are in the land where the dragons live and nobody can stop you. However, after some years, this is probably like year 2000 of OCT, 2000 to 2005. Then there comes the brute reality. There is this valley of death because all these ideas, or 99% of these world-saving ideas, do not work. And this is for quite some time. And many technologies die out here, I would say. We had examples like this over and over again. However, if successful, at some point it ramps up because when the technology becomes available, being commercialized, end users can apply it. Can be applied by doctors who know what they are looking for. And then, of course, output goes up again. And I would say somehow correlated with this, the benefit for society and the benefit for the patient. So overall, I would say, like technology, no matter how it cycles up and down, this probability of being accepted in hiring journal, if successful, the benefit of society is there. So don't forget technology. Thank you.
I tell you also a little story about this. Starts that you somehow early on have to take risk, and the risk taking here was very clear because we didn't know what to do to get on the visibility for inflammatory bowel disease, which was a topic I was working on. And so we decided basically that what we do needs to be something bold. And we decided that the way to do this, it's gone, the slide is gone, to be bold is that we do a clinical trial. And we did a clinical trial in a field where nobody else was under the way, which was at that time erythropoietin in anemia and chronic inflammatory bowel disease. So we did do actually three things. We correlated everything we have with everything we could. So we created IL-1 beta secretion with erythropoietin levels, two ELISAs. Then we found that erythropoietin is too low. We made our own statistics. There's no statistician on this. And then we decided to expose 34 patients, where at that time was a no-brainer. And then we decided to publish it, and I was more or less fired because I didn't put my boss on it. So I then went to Berlin and obviously created the appropriate visibility. So a splash at the beginning is always a good idea. And obviously times are different. This is back in 1996 when I was 34. The regulations were such that you could still do things like this. So let me go then on. Translate early. We thought that was a cool thing. And so we decided to measure IL-1 and IL-10 and look at IL-10 in different conditions, inflammatory bowel disease, laminar propria mononuclear cells. And you see how this is turned down and how this is downregulated. And then we created an enema with IL-10 and showed in a few patients that that worked. And that was also possible at that time, also 30 years ago. And it gave obviously another visibility. Okay. We thought we need to push on that way a number of times. And then another topic is use existing regulations. Obviously, more people try to do human experiments and skip animals. This is a famous one here. This is a microbiome transfer experiment or stool transplantation. This comes actually from a do-it-yourself page. How you do it, I hope we're never invited for a cocktail party in that household. But obviously, the reality how you do it is not so far different. And it's again an escape route of going from skipping animals and going to men, because again, look at unregulated territory now 20 years later. To do something with human stool until recently has not been seen as anything which is governed by any kind of drug law. So you could do whatever you like you can do lots of things with food, yeah? So you can also use stool. Now this is put under regulation rightfully, because some things have happened which were not so good. But I think for about 5 years or so, news are broken by just people doing that and skipping the animal. So that's the way how obviously this looks like inflammatory bowel disease. You see that there is obviously a signal, and that signal is obviously caused by a little bit cleaner way to use a stool transfer. Here you see this medical equipment or these capsules that are used. And there's still work going on in Germany of people doing this and trying to make science with this across the pharma, above or in the side of the pharma world. So we decided to ask the question, is it really necessary to have stool in a stool transplantation? Or that microbiome, is it really playing a big role? And so we said, let's filter this a little bit and filter that sterile. And indeed, if you filter the sterile, you get in Clostridium difficile a reasonable change of the microbiome, as you see here in the donor, the patient, and then the patient after that transfer here. That's just one or two of the patients. This was a little serious. Now, obviously, the world has changed, and this leads to fines. €500 for the first author, because now the regulators read the abstracts. Okay. Maybe you go a bit further. And Philip had already shown you that on the animal model here with the tryptophan. That's a discovery that has been made and that was explained to you, that shows very clearly tryptophan levels are essential for guiding the immune system, and if you take it away, you get chronic inflammation, and if you are putting it back in, and even if you do that in a normal mouse, you can combat inflammation. So what about patients? It's no problem to measure patients if you put the appropriate legal constructs in place, informed consent, ethics committee, whatever, and then you can start measuring patients. Wow, disappointing or not disappointing? That's a big question. I think it's not disappointing. These are humans. Humans don't behave like animals. They're not inbred. And so a considerable percent of the patient is below that, what you see in normal volunteers. So at that point, you go on, and if you go on and refine this further, then you see if you start putting the mediators into ratios with each other, that you indeed can pull the signals adequately apart and see a nice disease activity-dependent signal. And there's a rationale that comes back to the animal models. So you need animal models at the end, how this could work. And obviously, there's an immune pathway that exerts in the gut and that can push to IL-22 and that can obviously produce kynurenine as a T-cell immunoregulator, and that can also go into all kinds of other behavioral aspects. So by this point, I think we were smarter than we started. And the regulations have toughened up on us, and we thought we'd do everything right. And so we did this year. We went to the nutrition group, Matthias Laudes, myself, Claudia Schwarz, and the nutrition people said, oops, no problem. We do a nutrition solution for you. Nicotinamide you can buy in a pharmacy, 900 milligrams a day. Just go to the next pharmacy here. You can buy the tablets right away, and it's no problem. There's a daily allowance, which we haven't overcut. And then obviously shellac is licorice. Just put shellac around it. Do this whole thing on a nutritional machine and you're fine. So you produce a non-toxic product. Looks nice, and you can put this into people, and you see indeed this is a free nicotinamide. It's all absorbed, and if you put this now into this is a delayed release formulation, you bring it down to the microbiome, you can cross the duodenum and suddenly pump it out directly in the ileum and colon into the microbiome, and it stays there. It's not even going to the blood. This is 900 and this is 900. So this time, it wasn't a fine, but it was a letter. If they do any patient more, the fine would be bigger. €500,000 was the answer of the regulators. And we learned also something very important, for which we initiated a legal proceeding actually with our university. Indeed, you cannot use food in patients. If you use food in patients, you have drug. So things get ugly, and now we have to take time and obviously produce a tablet. And when you produce a tablet, we will get going at some point in next year to try it, and we have about 4 years delay with this. So final step into the humans. So final point, when you've done that, you may also convince somebody else to take the risks for you. So how does that work? So this is a therapeutic landscape in inflammatory bowel disease. These are two leading drugs, vedolizumab and adalimumab. I don't go much into what they do. There's an anti-TNF and there's an anti-alpha-4 beta-7 integrin. So if you have these two drugs, would you say this one is better than this one? Look, 16.9% versus 17%. Maybe first of all you say, wow, what is going on here? These guys treat their patients with something which has 17% efficacy. Go away. That's not the reality. It is the reality. As we have a disease with many, many unsolved issues. And then the maintenance, okay, this looks a little bit better. But would you really, on this here, bet the farm? Would you do such a trial where you run double-blind in 1,000 patients, these two drugs, and if you lose, you're out of the market with this drug? Yes, indeed, we convinced them at a weak spot when I think everybody, as it happens sometimes in companies, was just fired. And it happened that the two drugs are different. Then obviously you're also rewarded with a high-ranking paper. So these are little stories as points of interests. So what are the conclusions? Bringing findings early into humans is essential if you do disease research. Never forget about that. Don't create the number 10 animal experiment, still create it, but look for some collaborator, or if you're a physician, put your own efforts up to bring it into humans in a format which is legal, obviously. It's also a big learning experience. It gets tougher and tougher. You see the regulations, the critical mass increases, but I think it is possible. And then try to be out of the box. Because if you're young, you must be out of the box to replace political weight and critical mass, because there are older people like me who don't let you in, by the system, not by bad meaning. And you have to create your own experiment, even if it's small, and just publish as a surprise, and then you're recognized because you entered the stage. Thanks.
Well, thanks so much for the organizers for inviting me here to share these thoughts with you about. I didn't really want to talk about writing up because that's really on you. But what I want to do is tell you, maybe help you think about submission. And I'm an editor, so I couldn't help myself. And I think submitting a paper and learning about how some of the editorial processes work and what we're looking for in papers will ultimately help you to write your paper and to guide it into the right journal, whether that's a big name journal or not, as the case may be. Just as a little bit of introduction to Nature Medicine, and I also, we've heard about resilience, we've heard about team building and collaboration, we've heard about how long it takes to bring studies to fruition. And then I'm feeling really quite guilty now because I'm at the end rejecting papers. And I'm really sorry about that, but it's not personal. So we get around 3,000 papers submitted just to Nature Medicine alone every year. And you'll find that with some of the larger journals, I used to work for quite a large clinical journal, and we get about 10,000 submissions a year. So it is quite high volume at this level. And about 500 of those are reviewed, so around 15%. Now, this depends from editor to editor. I personally prefer to review fewer manuscripts, and then the percentage that I'm accepting is a little bit higher. I don't like to give people false hope and waste people's time by sending out a paper that I know ultimately that I don't want. But ultimately, around 5% of all papers that are submitted to Nature Medicine are accepted. Most journals, in fact, I should change the slide. We don't talk about acceptance rates. We actually talk about rejection rates. I'm not quite sure why that is, but it's quite disparaging. But we publish around 150. I think the most important thing to have success at any journal when you're submitting is to submit to the right journal and to be realistic about that. But what's most important is not necessarily the level of the journal, but the scope of the journal. So I do spend a lot of time rejecting papers, and it's not because I'm rejecting bad science. I'm actually rejecting papers that are inappropriate for the pages of Nature Medicine. Perhaps they belong in Nature Biotechnology, maybe they belong in Nature Methods, or even Nature Communications. But we have a very, it's quite a pain point for many authors, because we've actually also just gone through, sorry, I'm doing a shameless plug for Nature Medicine here. We've actually just gone through a major scope shift. In the past 2 years, we got a new editor-in-chief in 2017, the end of 2017, I think. And in the beginning of 2018, before I joined the journal, Joao made the choice to move the journal out of the preclinical space, doing fewer knockout mouse studies, fewer molecular genetic studies, and doing more clinical work. So now we're looking at clinical trials, we're looking at mechanistic studies in humans. So while we used to have a very big focus on preclinical work and translational molecular medicine, we're now much more interested in how that paper has an impact, as one of the earlier speakers was saying, on society and on the patient. It's a very patient-centric model that we're aiming towards. And the shift is slow. So you'll still see some of those older papers in the journal. But more and more you'll be seeing this change, and particularly over the past year. So each journal also on their website will probably have specific areas of interest that can help you submit to the right place. This is because we're a very general team, we've got some very specific areas of interest, and these change from time to time. This changes from editor to editor. Again, with my shameless plug, but all of the journals at Nature will have somewhere on their website guidance for authors that tell you what type of studies that we're interested in. For us, clinical trials are huge. We're very keen on those, but also any kind of these studies in humans. So I've talked a little bit about consulting the journal website and where you want to go with that. What's also really important is formatting and word limits and figure limits and things like this. When you're submitting, we'd like for it to not be a painful process for you. And we will never reject your article because it is too long, straight up. If we're interested in your paper, it's always the science that counts. You know, the grammar might not be great, you might not be within the word limits or the figure limits, maybe you've submitted a structured abstract. All of that stuff, that fluff on the side, it's never going to get a desk reject based on a format. And I think that that's really important. I've recently reformatted a paper for an author last week, in a bit of an emergency. And now I understand author pain, and I'm really sorry about all of the formatting. But eventually that will have to be done. But for your first submission, if you're gauging interest, I wouldn't waste too much of your important time on that. What we do need for peer review is enough detail of the experimental design in the main text so the editors and referees can decide whether or not we want to move forward with that. Along with this, in your pre-submission, particularly Stefan, for your clinical studies, we have some very strict requirements now. We are not familiar with all of the laws and the regulations. We assume that if you have ethical clearance, that you're abiding by the law. But we will check for ethical clearance in the paper. We'll also ask if it's a clinical trial for you to submit your consorting checklist, as well as your statistical analysis plan and your protocol, and that you've done all of the reporting guidelines as per the type of study that you're submitting. And what's also really important here is transparency of reporting, particularly in clinical reporting. So we do consider negative studies, but we consider transparently reported negative studies. So maybe you didn't hit your primary outcome, but you've got a lot of exploratory data that's molecular data that will inform the next trial and could inform patient outcomes. And so that to us is also really important. Once we accept a paper, in principle, we actually send a copy of the reporting summary to a service who goes and checks through every figure in your paper and lets us know whether or not you've included all of the right statistics and whether or not you've added all of those to the figure legends and to the methods. So in terms of scope and what we're looking for, I think when you're thinking about which journal you want to submit to, you really need to think about the appropriateness of the journal and who you want your message to go to. Do you want your message to go to bench scientists? Do you want patients to see this? Do you want regulators to see it? Do you want doctors to see this? So we do think about who our readership is and whether or not your paper is suitable for our readers. Also in this, when you're thinking about writing up your paper for a journal that is a general journal, the big Cell Science Nature, or even Nature Medicine, which has we like to think we have a general clinical and scientific audience. So they're educated, but they're not necessarily specialists in your field. So you want to communicate your message in such a way that it is understandable by someone who's not in the field. And there's a good chance that your editor's not in that field because editors by nature are non-specialists. We are interested in conceptual novelty. And this is different, and there's a different bar for whether or not it's a preclinical study and whether you need 17 figures with multi-panels, or if it's a clinical study, in which case you probably just need a really good primary outcome. And then we consider where this is and where it lays in the field. Is this a big advance or is it additive and like fills in gap of previous knowledge? Of course, we're looking for robustness of the methodology, the experimental design, and what's also very important is, are the conclusions strongly supported by the data that you present? More often than not, when we're asking people to revise papers, we're asking people to tone their conclusions down. Your data should sell itself. We don't really need you to sell the data to us because we can see through that. We read that every day. This is amazing. And then you look at the effect size, it's actually quite small. And the reviewers will see that too. And so I would say that's probably the number one comment we get back from reviewers about almost every study, which is the conclusions are aspirational and they're not truly supported by the data. So, but if the science stands, then we'll just ask you to rewrite that, and that's fine. And I think what's really important, if you make it into the 10% or so of submitted papers, that we would be requesting a revision. I think it's really important to know that what we need from authors is to engage in your papers. The purpose of peer review and the purpose of editors is really not to crush your life's hopes and dreams. I promise. We're really nice people. Our role is to help you present the strongest science that you possibly can in the best journal that you possibly can for your work. And so we're here, if we've gotten so far as to ask you to revise your paper, we're interested and we're on your side. What we try to ask authors not to do is to rebut everything. There's nothing more difficult than an author who reads a response to reviewers, is invited to review because we're interested in your work, and simply come back at every single point with the reviewers didn't understand what we were doing. If the reviewers don't understand what you're doing, then the editors don't understand what you're doing and our readers don't understand what you're doing. So if we don't understand, help us. Engage, rewrite the paper, call your editor, have a chat with him or her, and explain your points. You can ask for additional clarifications. Like I said, in the way that we divide our time, I would say that papers that are being revised, that's the point at which we have the most interaction with the editors. Once I've accepted it, it goes into production and you deal with other people. As it comes in, if I desk reject your paper, there's a good chance that we're probably not going to interact again about that paper. Then at this stage, we would ask you to go back to the formatting guide. And if we give you editorial points, please address those as well as the reviewer points, because if we do move forward that paper, it'll get you to accept and it'll help us publish your paper faster. So I'm always torn as to whether or not to use this slide, but people seem to really like it. So, and this is that you're always, and this is a dangerous thing for me to say, but you're always welcome to appeal my decision. And sometimes that can be really helpful. We make mistakes. Sometimes we choose the wrong reviewers, although hopefully not at the level of Nature Medicine. Sometimes the reviewers will be split and we'll have an editorial discussion, and we may not give you detailed information in that letter because we do have quite a large volume of papers to process. And maybe the reviewers didn't understand your paper. So on that basis, we do invite people, when they strongly disagree with that editorial decision, to come back to us with a reasonable point-by-point of how you would revise your paper and whether or not we would consider it on those bases. It's quite interesting because I find that a lot of authors read the reviewer comments in a completely different tone to what I would read them in. So someone will come to me with reviewer reports and say, we think this reviewer rejected it for this reason. But when I would read it, I would say, well, actually, it was rejected for loads of other reasons. So it's very interesting to see what people will pick up on in that sort of where your viewpoint for the work is. Where appeals are typically not useful is when you have an absolute unanimous conversion of referees that they don't like, that they're really not into the paper, particularly if many of them point out that the paper is not novel. And while you are welcome to appeal, we generally don't consider multiple previous versions of a manuscript. So if a paper was let go about 2 years ago, maybe you want to come back and revise and see if would consider it again. You may actually be better off using your resources and submitting elsewhere at that point. And also there's really no such thing as a sort of forcing your paper argumentatively into the journal. So this, I know that sounds like a real no-brainer to say, but we do actually get quite a lot of aggressive appeals, shall we say, which can be quite disheartening because you sort of already know what your response is going to be in that case. But if you only want to address some of the revisions and not all of them, then that's generally not acceptable to us either. So that is it from me. I know that was a bit of a laundry list of how we're doing, how we view your paper, but hopefully that's sort of a bit of a broad overview and can help with your submissions. And we welcome your submissions. I've actually been really, really impressed with the level of work that I've seen presented over the past 2 days. And you guys, the cluster is right in the sweet spot of nature medicine because we're very interested in this translational space and moving from the bench side into the clinic. So thank you for having me here. Really appreciate that.
Yes, so thank you very much, Jennifer. And so we have a little bit of time left for some discussions. And so I first like to ask the audience if there are some comments or questions that you want to ask.
Does it happen that you accept the paper despite of negative reviews, where maybe the technicalities have been accepted, but you feel you still want the paper?
Yes.
Ignore the, yeah.
I once accepted a paper with 4 recommended rejects. Every paper that's sent to review at Nature Medicine is discussed among the team with the complete set of reviews when it comes back. And we quite often have very split reviews, and it's very much an editorial decision as to which way we go. Like I said, one thing that I really like about being an editor at Nature Medicine is that we don't ask reviewers for their recommendations. So some journals will ask a reviewer to say major, minor, reject, or accept, whereas we only ask for your general opinion. We don't ask for your specific recommendation. And so it is up to us to measure that and see sort of what the general tone is. But also it has a lot to do with knowing your editor. And if your idea is really out of the box and you've discussed with your editor and you've convinced your editor, then there's a good chance that your editor is going to fight for you in that room.
So that means if somebody really has something out of the box, he better call you before he submits.
Yeah.
Oh, okay. That's a good point.
I mean, one of the main points as a scientist, right, is that you always have the problem. I feel scooped. There is a conflict of interest by the reviewer and something like that. I think every one of us, including you, probably has stories to tell where you felt that there was an unfair review process. So do you have, I think a good step forward is actually publishing the reviews. I think this is a really, at least for some of the reviews, very, very good step forward. But do you have any mechanisms how you follow up conflict of interest? I mean, stating no at conflict of interest is something that the person who has a conflict of interest and who has some criminal activity ongoing, definitely will not do. Yeah, but still there might be conflict. Do you have any mechanisms following that up?
Conflicts for reviewers? Mm-hmm. We don't have any specific, so that is one thing. Editors, we're not policemen. So, we have to take you at your word for what you say when it comes to conflicts. That said, each editor at Nature Medicine, we're all essentially integrated into our communities. So as the metabolism person, I go to a lot of diabetes meetings. And so I tend not to have people who I don't know review my paper. Well, I have at least one person who I know quite well and who I trust to review every paper that I send out. If a review is particularly scathing and it's out of sync with all of the other comments, there's a good chance that I'll ignore it. I will send it to the authors and I will say, these points have been raised, but it's out of sync with everything else. So please, can you, we would be fine if you were to ignore that. If a reviewer is particularly rude, and I've had some very rude reviewers, I wouldn't even pass those comments on to the authors. I would discount those immediately. I would consider them unprofessional.
Which is exactly the quality mechanisms that I meant.
Thanks. I was just wondering, I looked on the instructions for authors and I found that you actually offer pre-submission inquiries. So I was just wondering, what's your take on that? Do you recommend it or?
Oh, I like them. I think it saves everybody time. And this differs among my colleagues. Some colleagues would prefer to see the whole paper. Sometimes our standard formatting letter, when we get a pre-submission inquiry, actually says, well, yes, we're interested, but no, we can't tell you if we're going to send it out to review because we haven't seen the whole paper. And that is true, because sometimes you can get a really nice abstract and a paper that is possibly not quite as interesting as you had hoped. I personally, I'm quite happy to commit to reviewing papers. It's a fun part of the job. I don't mind reviewing papers. And I probably don't hit my 15% anyway, because I try to review papers that I'm interested in. So having these conversations with your editors, I think, are really important. I think I've just opened up all of editorial to extra work now. But for me, I actually find it much easier to tell someone really straight up, no, this is just not for us. And, you know, as long as you're okay with that and you can take it on the chin, then we're good. I think it's much easier to have that frank conversation, and I live in our manuscript tracking system, and it is a nightmare. It is slow, it is clunky, the links don't work. If I don't have to put you through that, I won't. Really, I think that's just an additional barrier to submission that we don't need to see. So pre-submission inquiries, I find them really helpful. You can submit them online. But you can also, if you know an editor, or you can see on our website that we've all got our emails there. And so you can choose the person who's most appropriate for your field and then put out a feeler, and that would be fine.
Yes, so thank you very much. I think time is over. Thanks to all speakers for very interesting talks, and especially thanks to Jennifer again for coming here and for the great discussion.